I don't know about you all, but when I think about metabolism, this is kind of what I remember. Flashbacks to undergrad biochemistry and cell biology. Oh my God, Krebs cycle, glycolysis, TCA, electron transport chain. When we talk about metabolism, this is what we're talking about. And when we talk about metabolomics, we are talking about using a technology to measure metabolism. So being new to sort of really diving deep into this subject, I started in PubMed. And so when you put in psoriasis and metabolism, you get a lot of papers starting way back when psoriasis started being explored in 1945. If you refine your search to psoriasis in metabolomics, we don't get papers until 2012, and we get far fewer. If you refine it further, and you specifically ask about psoriasis in immunometabolism, you get 10 papers. So the good news is we don't know very much, so I won't go very long. The bad news is I am going to talk to you more about metabolomics. All right, so as I started reading about metabolism and psoriasis, these are some of the lines out of papers that caught my eye. And here's the conundrum. Do changes in metabolism cause psoriasis, or does everything that's happening at the cellular level in psoriasis cause changes in metabolism? What is the connection? I don't actually think we really know the answer yet, which you'll see. So for those of you who don't remember, when you're talking about metabolism, it's really broken down into sort of 4 subgroups of biomolecules: carbohydrates like glucose, lipids like polyunsaturated fatty acids, proteins, and nucleic acids. So when we talk about metabolomics and the data that comes out of metabolomics, it's usually grouped into one of these 4 groups, which all plug into different metabolic pathways. So how does this work? So you got a great lecture from Johan earlier about spatial, single-cell, bulk RNA sequencing. Metabolomics is kind of similar in its process, right? You get a sample from a patient, and here's the difference. It can be from plasma, it can be from serum, it can be from skin, it could be from a punch biopsy, it could be come from a tape strip, it could come from a biogel put on the skin, and then you process what comes out of the biogel. You can use single cells, you can use urine. So there's this plethora of metabolism data in psoriasis from all these different types of samples. So you collect your sample, you can do a targeted or untargeted approach. It's, instead of having it sequenced, we do mass spec or NMR, and it plugs out a bunch of different data. Again, it gets analyzed sort of bioinformatically, it has statistics done on it, and then ultimately you get a pathway with increases or decreases. This is what the data looks like. If anyone's done metabolomics, you know the report comes out as a huge exhaustive list of changes in metabolites. And that's what you get. Maybe if you're lucky, they interpret it and they show you how these metabolites integrate into a metabolic pathway. This is how you can present it in a paper. And that's not complement, that's carnitine. So you've got increases in carnitine and spermidine and glutamate and methionine and putrescine and ADMA in lesional psoriasis skin compared to non-lesional psoriasis skin. And the authors of this paper said that these increases are reflective of cellular hyperproliferation, which we all know happens in psoriasis. So very, very descriptive, right? So what is known? What is known about the regulation? So these 5 review papers are kind of in obscure journals, but they are phenomenal. And the reason why they're phenomenal is because they literally give you every single metabolite that's ever been observed to be changed in psoriasis: skin, urine, serum, plasma. The list is exhaustive. And it looks kind of like this. There is a lot of data, but it's all descriptive. So you'll get a list. This is showing you a supplemental table from this review in Chinese Medical Journal from 2020. I'm only going to show you the list that came with glucose metabolism, but they also have ones for amino acids and nucleic acids and fatty acids. But you can see in peripheral blood, in this case, you're getting increases in lactic acid and decreases in skin, lesional skin. So this makes it further complicated, right? The changes are not overlapping between different samples taken from similar patients. And in urine, you get totally different metabolites found. So you're like, how do I integrate all of this? So here's a Venn diagram sort of showing you a summary of metabolites that are increased or decreased in skin, blood, and urine. And notice the overlap. There's overlap between polyunsaturated fatty acid oxidation, arachidonic acids, heats, leukotrienes that happen in all three of these sample types. But again, look at what it's linked to. It's implied for cell proliferation, cell growth, metabolite turnover. This is the level of knowledge that we have right now about the role of metabolism, metabolomics in psoriasis and psoriatic disease. So how does this work? So this is where we really are at this point. You're going to get a list of metabolites, the process and the pathway they're involved in, and how they're implicated. So how can we use metabolomics? Are we using it at all where it's useful, and where are we going to go? And how is the best way to use this method to gain knowledge into psoriasis disease, biomarkers, drug efficacy, drug responsiveness, drug non-responsiveness? I'm going to spend the last slides talking about papers that are out there which actually show something I think that's useful for psoriasis, and there's not very many. The first one is this paper that was published in Lipids in Health and Disease in 2021, and it's really focused on cardiovascular comorbidities in psoriasis patients. So what they did was they recruited a bunch of patients with psoriasis, and they did metabolomics. And what they showed was there was a bunch of different metabolites that were increased. When they treated them with IL-17A inhibitors, they decreased. Not surprising, right? And they then graphed it here. You can see the lipophosphatidylcholine, glycerylphosphocholine. As you go down and look, you can see increases in these metabolites or decreases, and IL-17 returned it to normal. And they show that some of these metabolites correlate with disease severity. Not unexpected, again, very descriptive. Next, they stratified patients with cardiovascular disease versus those without, and they did it based on coronary artery disease presence. And they also recruited another cohort of patients without psoriasis that only had cardiovascular disease, and they did the metabolomic study over again. And what they're showing here in this panel is this is the p-value change, and these are the metabolites that are increased between patients with cardiovascular disease and those without. And then they treated patients with anti-IL-17A, and again, they showed a normalization. So not surprisingly, what they derive from this is that dysregulated lipid metabolism is involved in cardiovascular disease. If you treat psoriasis patients with antibodies against IL-17A, the lipid metabolism sort of normalizes, and this suggests that if you treat the patient with this drug, maybe you will prevent or improve the comorbidity. So an application of metabolomics. What else? So as a preclinical person who does mouse model stuff, there's a lot more data looking at metabolism in animal models. Why? Because we can do things to mice that we can't do to humans. So GLUT1 was already talked about earlier today, and here it comes up again, resurrected. So GLUT1 is a glucose transport protein, and it moves glucose from outside the cell into the cell. Very simple. And you would assume that glucose metabolism is increased in psoriasis, and that's true because the cells are undergoing more metabolic changes. So this group did, in part by one of my colleagues, Jeff Rathmel, who is an amazing metabolism guy at Vanderbilt, took imiquimod, the Aldara model, an acute model of psoriasiform-like inflammation, and they treated mice that had either keratinocyte-specific knockout of the GLUT1 gene, or they treated mice with a GLUT1 inhibitor. And I think you can appreciate just looking at the epidermal thickness, the acanthosis, that if you knock out GLUT1 from keratinocytes, you get an improvement in disease. You get a decrease in Ki67 proliferating cells, a decrease in phosphosat-3. They show a bunch more in vitro data showing similar findings. And in the animal model, they also show that knocking GLUT1 out of the keratinocyte also decreases the number of T cells and the number of cells producing IL-17A, as well as number of neutrophils and the number of macrophages. So this suggests that skin inflammation occurs dependent on keratinocyte glucose metabolism, and that if you prevent a keratinocyte from having good glucose metabolism, you improve the disease. And the author suggested that maybe targeting glucose was a potential way for treating psoriasis. I'm not sure I agree with that, but it could work maybe if it was a topical. So what else? T cells. T cells that make IL-17 are critical for psoriasis pathogenesis. Here's another example where they took LAT1, which is also known as SLC7A5. It's a protein, it's a transport protein that allows large neutral amino acids like leucine to transport outside the cell into the cell, okay? And they knocked out this LAT1 transporter protein, again, from T cells this time. First time they used a ROR gamma T promoter to knock it out, which is gonna knock out the innate and the adaptive T cells. So the gamma delta T cells, which are really responsible for imiquimod-driven inflammation, but also CD4s and CD8s, all the alpha betas and all the gamma deltas. And they show, again, an improvement in the disease evidenced by decreased epidermal thickness. Then they did it with just a CD4 specific knockout. And again, they showed a significant improvement in the disease, and they showed significant reductions in IL-17A and IL-10. So this says that LAT1 allows leucine to come into the cell, and it actually has the capacity to signal through mTOR, which we've also heard about today, and it drives the expansion of T cells, the survival of T cells, and the release of IL-17A in synergy with IL-23. So in this case, metabolomics is helping us understand in the metabolism of a T cell. And together, they're driving psoriasis inflammation because we know the keratinocyte T cell interaction is so important for the disease. So where do I think metabolomics is going? So this study has limitations, but I love the concept of it because what it's doing is it's combining single-cell RNA sequencing to study metabolic change. And they're doing this by looking at specific metabolic pathways that, KEGG analysis will tell you about, and they're looking at changes in gene expression specifically related to these pathways. And I'm gonna focus only on this component. This is their graphical abstract, but I'm only gonna focus on one component of this. And I also wanna point out that they did this study using data that Wilson Liao originally published. They downloaded it from the cloud, and they reanalyzed it. So there's, for those of you looking for a unique niche, there's a plethora of metabolomics data that could be really creatively analyzed if you can write the code and do the analysis. Okay, so here's what they showed. So they specifically looked at T cells derived from PBMCs, and they clustered them based on markers of CD4, CD8, and Tregs. They also had mate cells and gamma delta T cells. But what I want you to focus on is these are the metabolic pathways they looked at, and these are the changes in T cells and then skin homing T cells. And the gray shading means decreases, and the white open labels mean increases. And what they found was in T cells, there was decreases in fatty acid degradation, as well as decreases in linoleic acid and arachidonic acid metabolism. Next, they took those specific fatty acid metabolites or metabolism pathways, and they aligned them with changes in gene expression related to these different signaling systems. And what they were able to show was that there was an inverse relationship with linoleic acid metabolism. Inverse, meaning decreases in linoleic acid in T cells, was inversely related to IL-17 signaling pathway, as well as Th17 and Th1 cell differentiation. So the less fatty acids, the more IL-17. Got it? And the same thing they showed in the Treg population. And so what they suggested was this audacious idea that supplementing polyunsaturated fatty acids might improve the disease. Now, I think it was Wayne who asked about fish. You know where this is going. So work that we did several years ago and that I have been remiss in finally getting published, we actually supplemented fish oil to our chronic model of psoriasis mice called the KC-TY2 mice. And like we literally just fed them fish oil, and we redid this experiment like 6 different times with 6 different people because I just didn't believe it. And the improvement in the histological phenotype is profound. I mean, they look like healthy control mice. And there was a significant decrease in the number of T cells as well as IL-17A production. So this sort of supports that data, combining the metabolic pathways and the single-cell RNA-seq with this hypothesis of supplementing PUVA signaling may improve the disease. Now, what's the mechanism? It's probably more likely that by supplementing omega-3 fatty acids, we are driving the metabolic pathways to an inflammation-resolving pathway rather than the omega-6 pathway that drives pro-inflammation. All right, so what's the future? You know where I'm going. You just heard Johan talk. We're going to go into spatial and single-cell metabolomics, and maybe this will be more helpful, right? What else are we going to do? We don't know very much. It's limited really primarily to animal models where we can manipulate things. It's more difficult to study changes in patients because it's harder to manipulate things, yeah. But I think where we're going is going into this multi-omics field where we can combine proteomics, metabolomics, RNA sequence, transcriptomics. I also did not talk about the microbiome. And the microbiome has its own metabolism that interacts with the human metabolism. And so that's another area that metabolomics might be helpful. And then also going to a systems level immunometabolism level, where we are studying the metabolism in individual cells and looking at how those metabolites interact with each other. That's going to give us more information about why things go up in one tissue, down in another tissue, and let's be honest, it's more likely that things are going up in one cell, down in another cell as they communicate. So I'm going to end as I began, which is a list of sort of sentences that caught my attention. And one of them is, this one. The metabolome is far more dynamic than the genome or even the transcriptome, which tells you that when you take your patient sample, you're only capturing that moment in time. The other thing is that metabolite identification is a foundational problem, as is the sensitivity of being able to measure metabolites. So I think as we move into the future, we're going to learn more about psoriasis and the role of the metabolism that hopefully will give us more pathogenic or resultant understanding of the role of it. And I'm going to end there and take questions. And hopefully I went fast.
Thank you so much, Nicole. Wonderful quick answer. I'm sorry. I mean, we tried to postpone the pickup by...
No, so you didn't allow me to ask a question about cure, but I'll ask a question about this. So this would probably explain why intermittent fasting works for psoriasis, because it diverts the metabolism down the free fatty acid pathway.
Right, so you're doing, not only are you diverting it to the fatty acid pathway, but you're probably also decreasing glucose metabolism.
Exactly.
So you're shutting down both the T cell and the keratinocyte and the interactions between the two.
Exactly.
Yeah.
And we have a presentation on that on Saturday morning.
Ooh, Gene to Clinic, don't miss it.